Chandigarh. Recently, I retired from Institute of Microbial Technology, Chandigarh, as the head of environmental science and microbial biochemistry. Now I'm working as a charity man. I'm director of Polyclinic, which is a hospital having many doctors. I do charity work and I'm the director there. And I'm also working with MBSI as the president of Chandigarh Tri-City. And I really appreciate the good work done by MBSI under the stewardship of Dr. Arvind Deshmukh. We have many dedicated people, and Dr. Deshmukh says less than 20% are dedicated. 80% they just watch, see, and go. So if we have even less than 25% dedicated people in MBSI, we will rise to the top. Isn't it, Dr. Deshmukh?
Yes, sir. Yes, sir. Thank you. Thank you very much.
Thank you.
Now, I will request Nikita ma'am to proceed. Nikita.
Your mic is off.
Thank you, sir. So, hi, everyone. I'm Dr. Nitika Thakur, and I'm working as Assistant Professor in the Faculty of Applied Sciences and Biotechnology, Shulani University, Solan, Himachal Pradesh, India. I welcome you on behalf of Microbiologist Society of India for this session, which is generally known as Sunday Talkies, and generally the series which belong to the Indians talking from abroad. So this is the 24th series which we are conducting. And I'm really glad to welcome our star of the today's talk, Dr. Supriya Gupta from Czech Republic. And she'll be talking about a very important topic regarding the exploiting the pharmaceutical potential of the plant endophytic fungi. Before we move on to this session, I would like to request our president, Professor Arvind Deshmukh, sir, to just share the vision and mission of MBSI. So over to you.
Thank you. Thank you, Nikita ma'am. I really appreciate your continuous efforts to arrange lectures of Indian origin, doing excellent job out of India. So today's speaker, I think her name is Shubhapriya Gupta. She is working in Czech Republic at present. Definitely, Indian microbiologists are happy to listen her because many young students are interested, have dream to visit sometimes in the life to Czech Republic. So today's lecture, as we are recording and we are circulating among 20,000 students, it will be beneficial. So I really thank Shubhapriya ma'am for accepting the invitation extended by Nikita ma'am and coming today on our platform. Thank you. Thank you.
Thank you so much, sir, for sharing the mission and vision of MBSI. So we'll move on to our session today. So we have Dr. Shubhapriya Gupta, who is a postdoc fellow in Laboratory of Growth Regulators, Faculty of Sciences, Pulaski University, Czech Republic. In addition, she holds an honorary research fellow position at University of KwaZulu-Natal, South Africa. Furthermore, she also works as director of Worcester Manuscript Services. She holds her PhD as well as master's degree from GB Pant University of Agriculture and Technology, Palampur, Uttarakhand. Her PhD studies highlights the characterization of foliar endophytic fungi as a promising source of bioactive metabolites. She adds to her name many national as well as international publications, books, editorials, special issues, popular articles, symposiums, conferences, and workshops. Her area of interest includes wide area related to biostimulants, seed physiology, endophytes, plant tissue culture, molecular biology, medicinal plants, plant microbial interactions, and analytical chemistry. Besides her active role in teaching and research, She also shares her immense interest in extracurricular activities. Without any further delay, I would like to invite Dr. Shubhapriya Gupta for her talk on exploiting the pharmaceutical potential of plant endophytic fungi. Over to you, ma'am, for your talk.
Thank you, Dr. Nikita, for the wonderful introduction. Thank you, Dr. Deshmukh, for all your appreciation. And it is my pleasure to present this talk. I'm so grateful for providing me this opportunity, and I'm more than happy to present this talk. So today I'm going to talk about exploiting the pharmaceutical potential of plant endophytic fungi. Dr. Nikita, just for the information, are there any students here listening to this presentation?
Yes, ma'am. We do have, and they'll be joining. Like if you proceed, and we have faculty, we have like researchers from globally. Yeah. So we have students also.
Yeah. Because mostly my motive was to just like maybe young students, young scientists, the budding ones.
Yes, ma'am. And your lecture is like live streamed on YouTube. So like many students have joined, and it will be streamlined for like 20,000, as Deshmukh sir said earlier.
Yes.
So
to all the students also who are listening, I hope that you might be interested on this topic, because this is very dear to my heart, this topic, endophytic fungi, because I did my PhD in this topic. And I find that there is a great potential in this topic, and there is a lot that can be done, because I think just this research is in the beginning phase, and there's a lot one can do. So to all the students and to all the faculty members of Shulani University and Ecological Society of India, thank you once again. So I would like to start this talk with a brief introduction about endophytes. So I think let's begin. Yeah. Dr. Nikita, if at any stage my audio is not visible or something is wrong, please let me know. So,
sure, ma'am.
Okay, so let's begin. So the endophytes, they are the microorganisms that lives inside the plant tissue in a symptomless manner, and they can be bacteria. I've shown here pictures of some visuals as bacteria, omycetes, or fungi. And these endophytes, they were first described by Freeman in 1904 in this English ryegrass, also called as Lolium temulentum or darnel. And we are interested in these endophytic fungi because they are a source of so many important secondary metabolites, having anti-cancer, antifungal, anti-diabetic, antimicrobial, immunosuppressant properties, and these are just a few. They present a lot of great opportunity and properties. So that's why we are interested in these microorganisms. And in this talk, I'm only going to focus about fungal endophytes, because they are easy to handle, and also they are easy to handle, they are easy to isolate as well. So with the students, like it can be really easy to deal with fungal endophytes as compared to the bacterial endophytes. So that's why I have only focused on fungal endophytes on this presentation. And these endophytes, they have been isolated from all sorts of plants. That includes angiosperms, gymnosperms, bryophytes, algae. They're even isolated from the plants residing in the extreme habitats such as mangroves, alpine habitats, and even from some desert areas. So we have around 300,000 plant species in the world, and it has been expected that there are around more than 1 million fungal endophytes. And for now, our scientists and researchers have already isolated more than 100,000 fungal endophytes. So you can see it has great potential. A lot of diversity is there. We just need to go out and find out the best ones. These endophytes also present so many significant novel opportunities, such as they can be used in agriculture, they can be used for landscape management, they can be used for pest management, and also pharmaceuticals, which is the focus of this talk. And as previously mentioned, that endophytes, they are able to produce the host metabolites. So first time in 1993, Sterling et al., they isolated one endophytic fungi, Texomyces indrene, here is the name, from a gymnosperm called Taxus brevifolia. And they showed that this endophytic fungi is able to produce Taxol, which is produced by the bark of Texas genus. And this is this was so interesting, and they published this paper in Science. And after that, so many other scientists, they also reported so many other metabolites produced by endophytic fungi, such as camptothecin, podophyllitoxin, vinblastine, vincristin, etc. And now let's talk about some bioprospecting strategies. Like, how can we isolate these endophytic fungi? What could be the criteria or rationale? So the first criteria could be random research, like we can select any plant from any habitat, and then we can isolate endophytic fungi for the potential secondary metabolites. Second is the ecosystem analysis, where we first study the plants and its ecosystem, and depending upon the interest, we can isolate endophytes. Third is the phylogenetics, where first we do the phylogenetic relationship among plants. And third is the traditional medicine, which is the most important and most used method for the bioprospecting of endophytes, because here we know that there are some medicinal plants, and then there could be potential endophytes which might produce the same host metabolite. So this one is the most popular method of isolating endophytes, traditional medicine. Now let's talk about the ecology of fungal endophytes. Just a basic idea, like how they reside inside the plant. So these endophytes, they can be, here I've shown in this figure, that they can be present in between the cells as intercellular mycelia, or they can be present inside the cell as intracellular mycelia, or they can be present both as intercellular and intracellular mycelia. Also, they can belong to all the fungal groups: Ascomycota, Basidiomycota, Oomycota, Zygomycota. Also, they have been isolated from almost all plant parts, such as leaves, stem, tendrils, seeds, bark, flower, roots, and fruits. So you can see, like, you can choose any plant part of your choice or any plant part of your interest to isolate these endophytic fungi. Now, the question is why we are interested in these microorganisms. We are interested because they are a source of so many novel biomolecules. That includes tannins, phenolics, alkaloids, terpenoids, lignans, resins, saponins, etc. We're also interested because nowadays there is a lot of deforestation going on, a lot of overexploitation of plants is going on, and that's why so many plants are, every year, so many plants are included in the endangered category, and there is a high market demand for the bioactive compounds. At the same time, there has been observed a decline in the rate of drug discovery in past decades. And that is why we are interested in these endophytes as their source of novel biomolecules. And we can mass produce these endophytic fungi in a bioreactors for the production of drugs all year round, irrespective of the environmental conditions. So the basic research outline of this kind of work is, first we have this is just for the students, like how we do the whole process. We do collect samples just as we do in tissue culture. We sterilize these plant samples, then it could be, here I have shown leaf, but it could be any plant part. And then we isolate endophytic fungi using various medium. Then we have to purify, because you see, like in single plate, you will be getting so many, 10, 20, even 50 or more isolates. You have to purify each isolate. Then we have to identify these endophytes with using molecular or morphological approach. Then we have to screen them for bioactivity. You can screen it for any sort of bioactivity that includes even cancer, Parkinson's, Alzheimer's, diabetes, all sort of activity you can test using the crude extract. Once you know that crude extract is showing some activity, then we are interested for the pure compound. Then we have to fractionate and purify the pure compound using crude extract. And then we have to do the chemical analysis. For chemical analysis, there are so many techniques that can be used. For example, spectrophotometry, NMR, LCMS, so many other, FTIR is also one of the chemical analysis that can be done. And then we identify pure compound. However, this process I have shown in the one single slide, it's really long, tedious, and we have to be very patient in this process. But if we are able to identify some drug or a compound, it can bring us a lot of, in research, you can publish your paper in nice journals, you can do something for the betterment of the society by doing this kind of research, because this research can be applied. It's not just the basic sciences, it's applied research, so it's very, very important. However, it requires a lot of patience and a lot of work is required for this. So here I have shown some structures of some compound which were isolated from 1993 to 2019 from endophytic fungi. There are so many other compounds as well. Here I have just shown 113 compounds. But irrespective of isolating so many compounds from these endophytic fungi, there has been very little commercial success. So that's the point of this talk, that how can we utilize this research and then, do some commercial success. So the limitations are that there are not many studies dealing with the ecology, physiology, and biochemistry of plant and its endophytic fungi. We don't know the exact biochemistry and physiology of plant and its endophytes. And also not many researchers are using advanced chemical, biotechnological, bioinformatical techniques. These are some limitations. And to discuss this in detail, I'm going to present here two case studies. The first one is of Taxol. And as previously mentioned, that Taxomyces and Rene is one endophytic fungi which was isolated by Sterling et al. In 1993 through Trixus brevifolia from the inner bark. And this endophytic fungi is able to produce Taxol. Because as we know that Taxus is the only genus which is able to produce Taxol, which is a multi-billion drug. But there are so many other endophytes apart from this Taxomyces and Rene, which is able to produce Taxol. Here, I studied like around 73 genera of endophytes which are producing Taxol. And these endophytic fungi, they are not only from the Taxus, but they are also from other gymnosperms. So out of 73, I studied like 51 endophytic fungi which belong to gymnosperms, and then 22 endophytic fungi, they belong to various angiosperms family. So this is really a very, very, a question can be raised that how can endophytic fungi, which is living in other gymnosperms as well as other angiosperm, is able to produce Taxol, because we know that Taxol is only produced by Taxus genus. And we would expect that only endophytic fungi residing in Taxus plants would be able to produce Taxol. But here we see that so many other so many endophytic fungi from gymnosperms and angiosperms are producing Taxol. So to understand this, we have to study the Taxol biosynthetic pathway. So in Taxol biosynthetic pathway, we can see that Taxol has been produced by this compound geranyl diphosphate through 3 molecules of isopentyl pyrophosphate and 1 molecule of dimethylallyl pyrophosphate through plastid-derived methyl erythritol pathway or through cytosol-derived mevalonic acid pathway. So 2 pathways are involved in plants through which they're able to produce this drug called Taxol. However, in endophytic fungi, the plastids are absent. So the only pathway is cytosol-derived mevalonic acid pathway through which endophytes produce Taxol. And also here you can see in this pathway, I have mentioned some enzymes in red color here. And some enzymes I have highlighted in black color, just I think 4 of them here, 1, 2, 3, 4, these 4 enzymes. So of all the studies done, scientists have reported these red color enzymes from endophytic fungi, but the enzymes highlighted in black color, they were not able to identify these enzymes or genes of these enzymes in endophytes. So what could be the reason? Like if these enzymes are missing from the pathway, how these endophytes are able to produce Taxol? So the answer to this could be that we are only able to identify the genes of the enzymes that share more homology. The genes present in the endophytes, which are most close to the plant genes of these enzymes, if they are close, then we are able to identify them. And if the genes, they share less homology or little homology, then we are not able to identify these genes. However, they must be present because we are still using plant-based primers. We don't have fungus-based primers for study of this enzyme. So that's why maybe we are not able to identify these genes. So there is a little ambiguity also, because the ambiguity is like some scientists say that maybe how do these fungus have these genes? What could be the reason how these genes have been transferred to endophytes? So some scientists have presented that maybe there is, during the course of evolution, there must be a horizontal gene transfer of all the pathway here, cytosol derived pathway. So, but it's really difficult that whole pathway has been transferred to endophytic fungi, because we see that the taxol biosynthesis, it consists of more than 20 steps. And all the enzymes are not only located in cytosol, but they're located in various intercellular compartments that include cytosol, plastids, endoplasmic reticulum, peroxisomes. So we can come to the conclusion that maybe the taxol biosynthesis pathway in endophytes are independent of taxol biosynthesis pathway in the plant, because it's really difficult. Like this whole, can you imagine like this whole pathway being transferred to the endophytes? So maybe they have evolved independently, just as we have gibberellic acid pathway independently in fungus. For example, there is this fungus called Gibberella fugiculae. It has all the pathway for the gibberellic acid biosynthesis, as well as plant, they do have their separate gibberellic acid pathway. So this could be the reason. And we are not able to isolate high yield of taxol from endophytes because we are not able to replicate the plant conditions. We are not able to replicate the exact light, darkness, pH, and temperature, like all the conditions which are required to produce taxol. We are not able to provide fungus in under exenic conditions, and that's why we are getting the low yield. Another major limitation apart from the low yield is the attenuation of secondary metabolites. Attenuation is like when we isolate the endophytic fungi, and then we have to subculture these endophytes in petri dishes or in slants. And when we subculture, we see that the yield of the secondary metabolites, it keeps on decreasing. So that's another problem. So to understand this, I'm presenting here the case study of Asiaticoside, which was my PhD work. And we were the first one to show that the endophyte Colitotrichum glisporoides, isolated from Centella asiatica, is able to produce its metabolite called Asiaticoside. This Asiaticoside has a memory enhancement property, and this is very important for for brain activity, and all this compound is very important. So we also isolated this plant, this endophyte from the leaf of the plant. And again, we had the very low yield, just as other scientists or researchers, they're experiencing low yield. And also we studied the effect of incubation time on the production of Asiaticoside, and we observed that at day 15th of culturing this enzyme in liquid culture, culturing this endophyte in liquid culture, we had like the highest yield, that was 62.29 micrograms per 100 mLs of media. And after that, it declined. And also when we were subculturing, so what happened here is we studied the effect of subculture generation on the production of Asiaticoside. And what could be what is subculture generation is like when we, when we subculture endophytes from one plate to another plate, like here I've shown in this figure. So what we experienced was, in the first plate, we had high yield, but when we were subculturing endophytes from one plate to another plate, because we have to subculture endophytes to, to maintain the purification. So we observed that when we were subculturing, there was a low yield. Here I have shown in this graph that the first subculture generation had like 62.29 microgram per 100 ml, but at generation 7, the yield was 4.26 only. And then at generation 8, it has stopped completely. So we could not found any Asiaticoside after generation 8. So this is the another second major limitation of this study. So to understand like what could be the reason of decline when we are subculturing the endophytes, what would be the reason of decline of secondary metabolites? So there are 3 hypotheses which are proposed by Sachin et al. In their paper. And I have also utilized this in my study. So the first hypothesis says that maybe the genes, they are silenced. When we subculture endophytes from one culture to another, the genes are silenced because we are not able to provide endophytes the exact plant conditions. The second is the fungal plasmid. So the endophytes, they have some extra chromosomal DNA called as plasmid, and maybe some of the genes of plant secondary metabolite are found is the fungal plasmid. But with each subculture generation, it is already known that the fungal plasmids are lost with each subculture generation. So this could be the second reason. The third reason could be that there are some endohyphal bacteria. So inside endophytes, there are some bacteria which reside inside the endophytes. And these endohyphal bacteria, they also have their own plasmid. They also have their own extra chromosomal DNA. And with each subculture, we also lose these endohyphal bacteria. And that is why maybe some of the plasmids are lost in the endohyphal bacteria. And that's why we have low yield, because we are losing genes of some steps. And yeah, there are so many studies. So we still need to test these hypotheses in this field. And there are so much other aspects to study, because not many studies are doing the elicitor study. So this is one of the future aspect, which I suggest that if somebody is interested, they can test some elicitors such as jasmonic acid, some hormones, some other elicitors, plant-based elicitors, to see if there is some increase in the production of secondary metabolites through these in these endophytes. Also, there is a lot of scope to study omics such as proteomics and genomics. Especially, we can add some CRISPR-Cas9 technology with this. However, this is very advanced techniques and requires a lot of money. But still, if someone has some funding, we can definitely try using genome editing techniques. And then maybe if some genes are missing, we can isolate those genes from plants, insert them into the endophytic fungus, and then maybe get the higher yield. Also, the another problem with this study, the another shortcoming is if there is some novel molecule, if we have isolated some novel molecule from the endophytes and it has never been tested, so there is a lack of preclinical and clinical trials. And because it requires a lot of funding, so the best thing would be to, collaborate with some pharmaceutical companies so that you can get some funding and do some clinical trials. So this is a very emerging field. A lot of things are still to be done. However, it's very interesting, very useful, very applied. And if some students are listening, you can also consider this kind of work for your master's or for your PhD. And that was all for today's presentation. And I would like to acknowledge my University of KwaZulu-Natal, Pulaski University, Czech Republic, and also Gustaran Manuscript Services, because their editors, they helped us in, doing this paper, and we published this paper in Biotechnology Advances, which is having a very high impact factor of 14.8. And if someone is interested, all this, what I have presented today, is written in so much detail in this paper entitled A Critical Review on Exploiting the Pharmaceutical Potential of Plant Endophytic Fungi. If some students or if some faculty, they have some questions or discussions about this, just go through this paper and then you can discuss it with me, and I will sort out all your queries if I am able to. And thank you again for this wonderful opportunity. And I really enjoyed presenting this talk. And thank you very much for your patient listening.
Thank you so much, ma'am. So it was a wonderful talk and an insight towards like the endosymbiotic theories and practical implications. So the house is open for discussion, and I request everyone to march forward with their queries. If you have any queries related to the topic, you can just message in the chat box, or you can just unmute yourself and ask.
Okay, then I will ask one question.
Yes, sir, please proceed.
Yes. It was a very good talk by you. Very nice.
Thank
Really
you, sir.
enjoyed the talk. It's a very modern talk using instruments, high throughput instruments. So, did you work only with fungi or also bacteria?
No, sir. I have worked with bacteria, but in detail, I have only worked with endophytic fungi.
I see.
Yes.
So, what do you find in case of bacteria? Are
No,
they
actually,
better or like fungi?
no, actually, the good thing with bacteria is that you get yield so fast, because you get the new generation of bacteria just in few hours. So even we can do the study so fast. The study with endophytic fungi is quite slow because you have to subculture or culture the endophytic fungi for like 15 days or 21 days. But with bacteria, we can get results in just 24 hours or 48
Excellent.
hours.
Excellent.
Yeah. But the thing is that in bacteria, what I was facing difficulty, like they're easily contaminated. And yeah.
In higher temperature, maybe
Yeah.
bacteria will do better than fungi.
Yes, I agree with you. Yes.
You should also concentrate on bacteria for high temperatures.
Yeah, you are so right, doctor. But I only worked with endophytes because the good thing with these endophytes, endophytic fungi, is that they are not easily contaminated. If there is some contamination, I'm able to see the contamination immediately. But with bacteria, it was so difficult. And like I did this work in PhD, and Indian labs, they are not very. Some of the labs, at least, are not very high end labs, and we face a lot of trouble. So my only agenda was to work with endophytes just to avoid that contamination stuff. But if someone is having good facility, good lab, I would definitely recommend working on bacterial endophytes because they are, yeah.
Okay, thank you.
I would like to ask one question, ma'am,
Sure, sir.
which is I will consider as a sub question to
Sure.
Sir what he has asked. I want to ask you, or I want to tell you, actinomycetes.
Mhm.
These are similar in morphology to the fungi, but they are bacteria.
Yes.
Correct?
Yes.
So when Swarnajit Singh sir have asked you the question, have you studied on bacteria? I would like to add one question that what is your opinion regarding endophytic actinomycetes?
Yes. Dr. Deshmukh, thank you for this question. As
Yes.
I said, that the bioprospecting totally depends upon in what we are interested. It's not necessary that we have to work on actinomycetes. It's not necessary we have to work on bacteria. We can select any endophytes. We do have endophytic viruses as well. We can work on endophytic virus. We have actinomycetes, we have oomycetes, we have all sort of bacteria, all sort of fungi, we have all sort of viruses present. Even some other microorganisms are also there inside the plant. So it depends like in what we are interested, because as I said that there are almost more than 1 million endophytic fungi only present inside the plants. And the diversity of bacteria is more. So we can expect there are more than maybe 1 million, 2 million kind of bacteria. So it totally depends on what we are interested. It's just up to the researcher on what they want to do. For example, if some plant is, if I'm interested in some plant which is in a swampy place, I might expect that maybe it has more bacteria. And if it's a desert plant, it might have more fungus, because in desert plant, desert plant, they do need mycorrhiza, they do need fungi for the uptake of water. So it totally depends on what's the ecology of plant, what's the environment of the plant. And then on that basis, I can select that if I'm interested in bacteria, if I'm interested in fungi, if I'm interested in ectomycetes or virus.
Okay, thank you.
Yes.
Thank you, ma'am.
Thank you. Thank you, Dr. Deshmukh.
Yes.
Thank you so much, ma'am. And I have a query, like you just mentioned about the commercial issues and flaws, which are generally related to this typical field. So are there initiatives you are sorting out or you are figuring out to tackle with these commercial issues?
No, Dr. Nikita, thank you for this question. I think my group, they are working on this, but currently as I'm working on biostimulants, I mean, they are also one kind of biostimulants can be utilized in agriculture, but yeah, the focus of this talk was only pharmaceutical, pharmaceutics or pharmaceutical kind of stuff. But currently I'm not working on this because right now my focus is on agriculture. But my group are still working on genome editing, but maybe they will find something useful in future, definitely.
Thank you so much, ma'am. And anyone who wants to go for the queries and all the like your questions related to the topic today? Any student, any faculty, any researcher who wants to come forward with the query? So we have a message in the chat box, ma'am. So thank you, ma'am. We'll go through the paper definitely. So this is from Dr. Yamini Dixit.
Sure. So yeah.
So thank you so much, ma'am. So,
yeah. I'm again highlighting the paper. You can have a photo of this paper, and then it's a very interesting paper. Like you'll find all sort of details. And I'm sure that if some student or faculty, they read through this paper, they will have all the answers, because I have covered not only ecology, physiology, biochemistry, all the biotechnology associated with this work. It's a very big paper, like more than 26 pages or 24 pages. And I really believe that you'll find it very interesting, because this is one of the best papers of our group.
Thank you so much, ma'am. So I think nobody has now any additional queries regarding it, and they'll be definitely going through this paper. And this seems very exciting.
I just want to ask you
sir.
something.
Yes,
You mentioned about biosurfactant, right?
No. Biostimulant.
You did not mention about biosurfactant?
No, sir. I mentioned biostimulants.
Okay, I thought you mentioned about biosurfactant.
No. Biostimulants.
Biostimulant also is a very interesting field.
Yeah.
So you should pursue that biostimulant.
Yeah, currently I'm working on smoke-derived biostimulants and CV-derived biostimulants.
Right.
And yeah, so this is the area right now I'm exploring.
This actinomycetes, can you give me a few examples of actinomycetes?
No, sir, right now, no, because I've never studied, I've never worked on any actinomycetes. I've only worked on endophytes. So I'm sorry.
Just telling you.
Yeah.
You can work on arthrobacter.
But sir, actually, I'm not working on actinomycetes, and also.
No. I'm just telling further to Dr. Deshmukh's question.
Sure.
Examples of actinomycetes are arthrobacter.
Okay.
Streptomyces.
Yes.
Okay.
Yeah.
They not only do stimulants, but they also produce very good antibiotics.
Yeah, sure, sir.
All the actinomycetes are gram-positive rod bacteria.
Sure.
They make a lot of antibiotics also.
Yes.
Okay, thanks.
Thanks, sir.
Thank you, ma'am. So we have, I think, a list of queries here. We'll take one or two. So
sure.
This one is from Sunita. So Sunita ma'am has asked, like, do you think once we isolate the organism from its host, it is still able to produce metabolite?
Yes, that's what we presented here. You can see that we isolated this Coleotrichum. This was my PhD work. So I isolated this Coleotrichum glueus. I mean, not only this, but I isolated 7 other endophytes as well, 7 endophytic fungi from the leaves of the Centella asiatica. And in exenic conditions, in the in vitro conditions, it was producing Asiaticoside. Here I have cited this paper below, Gupta et al. 2018 World Journal of Microbiology and Biotechnology. You can go through this paper. And you will find out that how we are able to find out that this endophytic fungi is producing Taxol. And I also mentioned that at the first generation, it was producing highest amount of Asiaticoside, but after 7th generation, this completely stopped. So yes, at some point, like after so many subculture generation, after 1 year or 2 year, you will see that your culture is not producing Asiaticoside or any other secondary metabolite, but yes, they do produce. We have to find out some ways. That's why I mentioned here that maybe we can use some elicitors or some other technique can be used. We can use some plant extract with the culture media to see if the production is stable or it's increasing. So the only problem is that the secondary metabolite production, it reduces at certain point, it stops completely. But yes, these endophytes, they do produce plant metabolite.
Thank you, ma'am. So second question is from Nirmala, ma'am. So she has a query regarding the yield. As you mentioned that yield is less. So she is like inquiring, okay, can we use a protein designing for this?
Yes, but for that, we have to be very specific. We have to first...
Asha?
Ma'am, just go ahead, please.
Yeah, so sorry, ma'am. So, we have to first know like which protein is not present or which protein is less productive. So for that, we have to study the whole pathway. We can only design the protein if we know there is a lack of certain protein. If we are not aware of that, we can't just produce any random protein. But if we study the pathway if we do the enzymatic study, then we can know that this protein is absent or is under expressed, and in that case we can definitely do.
Thank you, ma'am. So we just need to hit the target. We need to go for the target.
Yes.
So everything starts with the target, and research is all about target.
Yes,
so definitely. We have third question, ma'am. So Rupa, ma'am, she is like interested in knowing any promising application from your lab or from your studies.
Okay, promising application is like the only promising application we got, that we were the first one to find out that endophytic fungi from Centella asiatica is producing Asiaticoside. We were about to file a patent for this, but we could not do it because of certain things, because we already published that paper. We were new in the field, and then we didn't know that if you already publish your paper, you can't file a patent on that. So that's how we missed our patent. But now, like it was like 7-8 years ago when I did my PhD in 2015. So I was not aware at that time that we could file a patent, and we have to file a patent before publishing a paper. So that was the only limitation. Otherwise, we were the first one, and we could have easily got this patent on this. So this was the best part.
Thank you, ma'am. And the last question. So it's like, could you throw some light on postdoctoral opportunities or visiting your lab?
Sure. There can be a lot of opportunities, and those who are interested in postdocs, Dr. Nikita, you have my email ID. You can maybe send your CV directly to me, or maybe you can have a chat with me on my Gmail, and we can discuss about because sometimes we do have some positions, or maybe, for example, I'm collaborating with a lot of other laboratories in Europe and South Africa, in US as well. So if there are some position, I will definitely let you know. It depends upon which kind of field you want to work, because I'm working on biostimulants, and so most of my group are working on biostimulants, but yes, also plant tissue culture, also endophytes. So we do have some positions in plant sciences. Definitely you can share your email ID, and then we can even discuss the potential of having a postdoc.
Thank you so much, ma'am. So it was wonderful hearing you, and we'll invite you for the further talks also, future talks, and sharing your research. Yeah, and your target areas and all. So I think, yeah, ma'am, Sabha ma'am, we'll be sharing Supriya's ma'am's ID on the group. So I'll be sharing that with the PPT. So we'll share that, and you can collaborate further for your research proposals and all.
Sure.
So thank you, ma'am. Thank you. It was great listening to you. And I would like to thank everyone here. and mainly our Professor Deshmukh sir, Professor Swaranjit sir, all the teachers, faculty members, and the most important audience, our students as well. And our speaker, so a session speaker was wonderful. I think everybody enjoyed the talk, and we'll be waiting for her target analysis and all the, she comes up with all the type of future talks, and we'll be communicating further with the research collaborations and all. So anybody who is interested in collaborating with Dr. Subhapriya, he or she can generally ping her up on her mail, and I'll just be sharing her PPT with the mail. So anyone who has the similar research interest can easily collaborate. So thank you so much, ma'am. Thank you, everyone. So keep growing, keep glowing. Take care. Bye-bye.
Thank you. Thank you, Dr. Nikita. Thank you, everyone.